Welcome to Cannabis and Evidence-Based Medicine, where Dr. Russo is going to tell us about the standards of evidence required for cannabis to be considered a safe and efficacious medicine for a given indication. He's also going to tell us about which indications we already have strong or intermediate evidence. Dr. Russo, please take it away.
Thanks, Nisha. So as usual, here are bios, which you can study later. So the objective of this module is to explain the terminology of statistics employed in cannabis clinical studies, then go on to address the placebo effect, the regulatory process for pharmaceuticals, and to provide the status of evidence for or against cannabis efficacy in a wide variety of conditions. So first, some definitions: average, median, mean, and mode. The arithmetic mean is an average, as most people would understand it. So adding up all the values, their sum, divided by the number of values. And this is what people usually think of as an average. The median is the middle value that separates the greater and lesser halves of the data. And the mode is the most frequent number encountered in a sample, although it isn't often cited as being particularly important compared to the others. You'll hear a lot about standard deviations. That's a measure of how much variation there is from the average in a set of data. The lower this number is, the greater the consistency in the clinical results. A figure often reported after the mean is this. So you'll see 35% plus or minus 2.5%. And the 2.5 would be the standard deviation. Very important in epidemiological studies are relative and absolute risk. So the relative risk reduction describes the effect of an intervention in relative terms between two groups, and the absolute risk reduction describes the effect of an intervention in absolute terms, so it's typically a lot lower than the relative risk ratio, as it takes this into account. Risk ratios are the risk in a treated group over the risk in the control group. So the risk ratio compares the risk of a particular event in the treated group compared to that of the control group. The event can be good or bad, and risk ratio less than 1 means that the risk is lower with treatment or whatever the effect is that's being studied. Probability or p-values. A p-value of 0.01 means that there is a 1% likelihood that the observed result is due to chance. So that would seem unlikely. For medical efficacy, the real cutoff point is 0.05. So that or less is statistically significant, and is taken as the benchmark of medical efficacy. In contrast, a p-value of 0.01 or less is highly statistically significant, and a p-value of 0.001 or less is very highly statistically significant, because that would equal a 1 in 1,000 chance that the result is not due to whatever you think it's due to. Confidence intervals. These provide a range of values that one can be fairly confident that the true value lies within that range. By convention, with a 95% confidence interval indicated that for a given range of numbers, it is 95% certain that the true value lies somewhere within the range. Parametric and paired. Parametric data follows a normal pattern of distribution, and this applies to many but not all biological phenomena. Data are said to be paired if they consist of 2 or more measurements taken from the same subject. Number needed to treat is very important in terms of medical interventions. So that's the inverse of the absolute risk reduction. It indicates how many people would need to be treated with a particular intervention in order to prevent one event. So say how much how many people who get an antibiotic are needed to show that it had benefit in treating the infection. A corollary is the number needed to harm, and that's often applied in studies of cannabis that are meant to show its side effects. Beyond this, you really need a statistician. And if you feel lost already, stats are something that really confounds a lot of people trying to evaluate studies. Now, let's shift into assessment of what medicine is about in terms of proof. I like to call this the four pillars of true medicine. To show that something is a medicine or a good medicine, you have to show that it works, it's efficacious, and that it's safe. Also, for pharmaceutical approval, you've got to indicate that it's standardized. It's showing a high degree of consistency over time. And I add a fourth thing that's not commonly put in the writings about this, and that's accessibility. So this would encompass the medicine's availability and affordability. So if you have a drug like a biological that costs $100,000 a year and somebody isn't insured and doesn't have access to it, it's certainly not going to do them any good. So I think this is something that definitely needs to be taken into account. The levels of medical efficacy run along these lines. And let's go from the bottom up. Um, Level 5 are case series or studies with no controls. And at best, that may be what a lot of cannabis literature shows. Above that, level 4, historical cohort of case control studies, then 3 cohort and case control studies. Occasionally you see these with a variety of different types of cannabis, but a big problem in the field is just asking people if they use cannabis to treat a given condition. You know nothing about what was in the material. Was it THC predominant? Was it CBD predominant? Was it smoked? Was it ingested? So it makes it very hard to sort any compelling data from that. To some extent, with cannabis, we have small randomized controlled trials with unclear results. Better than that, obviously, are large phase 2 or phase 3 randomized controlled trials with clear-cut results. And the only place we have that is for synthetic THC, for Sativex, which is an extract of a high THC and high CBD extract that is administered oromucosally for treatment of spasticity and MS, among other things, and for Epidiolex, um, cannabidiol extract that is FDA approved. Beyond that, there's another level on, you will see studies in this regard called the meta-analysis. So this takes into account observations from multiple studies. The positive effects can be much more dramatic than those from single studies, and they carry a lot more weight. Similarly, the p-values are much better in general. Randomized clinical trials. This is the sine qua non of medical proof. In other words, the benchmark that you have to attain. This comes in 3 phases, or actually 4. We'll mention that in a second. Phase 1 is testing the drug in normal people to see how tolerable it is, get an idea what dosing should be like, and its pharmacokinetics, how much is in the blood at a given time. Phase 2 is where you test the same compound in a specific disease or condition with smaller number of patients and again assess safety and efficacy. So an example there would be a couple of trials of cannabidiol for schizophrenia, but these haven't gone on from there. Phase 3 is testing in a condition with large numbers, again, to assess safety and efficacy. Again, examples there, nabiximols, Sativex, THC CBD mixture with other components for spasticity or cancer pain, at least in Canada. and CBD is Epidiolex for seizures. Phase 4 is after drug approval, post-marketing surveillance or other studies. For example, if a drug were approved for spasticity and then you wanted to get it approved for a separate indication like sleep improvement, that could be a phase 4 study. Standardization. This is evidence that the drug is consistent chemically and identical in structure and dose over time. This is a real challenge for botanicals, drugs derived from plants, but it's been previously attained. How about pharmaceutical cannabis-based medicines? Well, like any medicine going through the regulatory process, it needs to be standardized and consistent with quality equal to any new chemical entity. NCEs are any synthetic drug like those that have been the model for the last 75 years. You must possess a practical and suitable delivery system that minimizes patient risk. And particularly for cannabis, this would include intoxication. You have to show whether it's got drug abuse liability. Do people like it too much? Do they become dependent? Do they have withdrawal? Or any other serious adverse events, such as hacking cough with smoking. Cannabis-based pharmaceutical must also have a supply chain that ensures security, that is being distributed to its intended target patients without diversion. I was always amused in Canada when medical cannabis was first legalized. This was shipped in a box to patients with the skull and crossbones prominently displayed on the front. Additionally, cannabis-based pharmaceutical must be accessible, again, meaning available and affordable. Why would it be an advantage to have pharmaceutical cannabinoids as compared to just having run-of-the-mill cannabis that people either make into brownies or smoke? Well, it is because these can be proven safe and effective as pharmaceuticals. And for most physicians, that's the only thing they're going to accept. These would be preparations that physicians can prescribe. They can be confident in that they work and that they're safe. Also, that pharmacists will endorse and supply. Also, prescriptions are the ones that government health services in this country, Medicare and Medicaid, and third-party payers will cover. They're very unlikely to cover herbal cannabis, although that has been the case in a couple of European countries. Why does black market cannabis fail these kinds of challenges? Well, it can't gain regulatory approval in most nations. There's a tremendous biochemical variability in the chemovars. Again, it should not be called strains, which applies to virus and bacteria. Again, unregulated material may pose risks. These can include molds, coliform bacteria, pesticides, or heavy metals. Smoking additionally imposes risks. Chronic cough, phlegm production, bronchitis, and inhalation of pyrolytic byproducts that are possibly carcinogenic, although THC and other cannabis components seem to reduce the risk of cancer related to smoking when it's cannabis alone and not with tobacco. Cannabis inhalation produces a rapid peak in serum and brain concentrations. That tends to maximize intoxication. It also can lead to reinforcement on the person wanting more. And those are definite risk factors for drug abuse liability. So a slower onset of action, like with an oral medication, is going to have a smoother contour, fewer peaks and valleys, and much less likely to lead to these kinds of issues. We need to take into account what patients really want. If someone has chronic pain, they want it relieved. Similarly with other symptoms, and they don't necessarily want to take a medicine to alter their state of consciousness. Now, I'm not going to tell you that euphoria is a bad thing, but it is to the Food and Drug Administration because if a patient is euphoric on the drug, that's listed as a side effect. And it also is going to create concerns about drug abuse liability and lead to tougher scheduling, meaning it is more controlled or harder to get. Patients also want a medicine that's consistent in quality and effects, and one that they can obtain legally, they can get to, and that their doctor recommends. Now, all of this, the drug development process for a medicine derived from a plant, is contained in this document, the FDA's Botanical Drug Development Guidance for Industry. Now, if you look at the items in it, it's the same as for a new chemical entity. As we've discussed, the quality, the safety, clinical data based on several hundred patient years of data, strict reporting of all adverse events, etc. The difference comes about in the characterization of the drug. And for a new chemical entity, you're usually dealing with one compound, and if it is present in each batch within a tight range of concentration, you're home free as long as it's safe. For a botanical, you may be dealing with hundreds of different components, and so it is that much more difficult to demonstrate the consistency over time. Now, I don't have that slide. I should have included here, but I could show you what happened with Sativex. Over the course of 9 years, 25 different traces of concentration of components, and it looks like single peaks. So that level of control of the ingredients is what's necessary for a botanical. There are additional Food and Drug Administration regulatory requirements. Again, detailed pharmacokinetics that we've mentioned, also pharmacodynamics. What's the effect on people? Does the drug increase or lower blood pressure? Does it get them high? These are all considerations. We have to have monitoring for contaminants. Um, uh, all the new drugs need to have studies in humans of effects on the electrocardiogram, what's called the QTc interval, to ensure that that isn't unduly prolonged that could cause dysrhythmias in the heart. It also requires human drug abuse liability studies if there's any indication of its psychoactivities. And it needs to be studied in patients that might have kidney or liver disease and have greater challenges. Very quickly, the available cannabinoids consist of dronabinol, the generic name for synthetic THC, trade name Marinol. This was approved by the FDA for chemotherapy-associated nausea in 1985 and placed in Schedule II, which, aside from the most dangerous forbidden drugs in Schedule I, means that you can only have a written prescription each month and not have refills that the patient can access ad lib. In 1992, it was also extended to treatment of HIV/AIDS wasting. And in 1999, it was Schedule III because there had been no particular diversion of this oral medication. There was no black market for Marinol. Also in 1985, Nabilone, which is a semi-synthetic analog of THC, was also approved, but it wasn't marketed for many years and has never had a good market share. In contrast to THC, it's a full agonist at the CB1 receptor. It's about 10 times more potent than THC, and correspondingly is more prone to side effects. Then we've got nabiximols, the trade name for Sativex, the oral mucosal preparation that's approved in 30 countries for spasticity and multiple sclerosis, but also central neuropathic pain in MS and treatment of opioid-resistant cancer pain in Canada. Then Epidiolex, again, 97% pure cannabidiol as an anticonvulsant for severe epilepsy syndromes. That was approved by the FDA in 2018. And there might be various others depending on the laws and jurisdictions. An assessment of how cannabis research works or doesn't work in this country. Until very recently, all the cannabis used for medical research came from the National Institute on Drug Abuse. Currently, there are 8 companies that hold DEA cannabis production licenses, and many of them are scrambling to find a market for their products. Technically, no other domestic materials permitted, even if it meets standards of good manufacturing practice, except perhaps for observational studies. And this is due to at least the interpretation under international laws, Schedule I restrictions, and then again, how the American government interprets the Single Convention Treaty of 1960. Clinical trials require state approval, institutional review board or ethics committee approval, and most often also need Food and Drug Administration investigational new drug application approval, which is a very involved procedure. Between 1999 and 2014, there was an additional Public Health Service review in the United States, which has been eliminated, and it really hasn't increased the number of studies that have been done. All studies to date with cannabis, which have been primarily in California, have been small in scope, short in duration, and basically are unreproducible due to lack of availability of standardized material. Now, let me expound on that. The material from NIDA was never standardized. Additionally, it does not resemble cannabis that might be available either in dispensaries or in the black market, often having cannabinoid content that's very low, and a dearth of terpenoid concentration. If, for example, you did a study with NIDA cannabis, it would generally be smoked, and you showed that it helped treat pain, low back pain, just as an example. Unfortunately, you would not get the same material again, and it would be impossible for this study to be reproduced. Now, this is going to sound cynical, but it's true. Things were set up this way. It's a system that was designed to fail, and in that has been very successful. There remain many federal roadblocks to cannabis research. The rules were created by successive administrations that, for the most part, have been totally opposed to the idea that cannabis could be medicine. In federal law, you will still see the term marijuana with an H. That's unscientific and pejorative. The name is cannabis. I will say that I do not think that the Food and Drug Administration is the problem. This is very much an organization that plays by the book. So if you're trying to prove that you're good for spasticity, you have to meet or exceed what the last guy did in terms of numbers of patients treated and the kinds of measures employed. As it is, cannabis remains a Schedule I forbidden medicine, which was never true when it came about through the Controlled Substances Act in 1970, and it's not true today. So depending on the state, doctors of whatever stripe, nurse practitioners, and sometimes doctors' assistants act as gatekeepers in the legal states, but they may not have a background to really understand. And there are vast disincentives on most instances. For example, in Canada, where cannabis has been legal in a form or another for more than 20 years, there's a single malpractice provider that actively discourages physicians from taking part in the program. The information that's available to consumers on chemovars is totally inadequate. People go by strain names, which are meaningless. There's no consistency from one place to another. Um, and often one only learns the THC and or CBD content without minor cannabinoids and without terpenoid profiles, let alone testing for dangers from pesticides, etc. Many patients are leery of the restrictions and would prefer to grow themselves. Now let's talk about the placebo effect, which is of great import in cannabis-based studies, at least for preparations that are psychoactive. It is the case that the mere act of being in a clinical trial generates a certain degree of improvement. Um, Patients are optimistic that they're trying a new thing and that's going to help them. Now, that certainly can be aggravated when there isn't a way to objectively test what's going on with the patient. We don't have a pain meter that you can stick on the patient's forehead and know what their pain level is. We need to ask them what it is. The situation is also aggravated when the drug is psychoactive. So this would apply to antidepressants as well as cannabis. And a real killer is if people get the impression that the drug is miraculous, and certainly people may feel that way about cannabis. Unreasonable expectations lead to unreasonable results. Everybody will hope that they're on placebo, and many will state that they're improved when it may be the case that there isn't a good physiological or biochemical reason that they're better. Now, the situation has become worse over time. This was a very sobering study in 2015 that showed increasing placebo responses over time in this country in neuropathic pain studies. And there was a very highly statistical significant increase in placebo responses over the course of 23 years. Um, While drug responses in early studies decreased an average of almost 35% from baseline and were stable over time, um, producing 16.5% greater pain control than placebo, or 1 point decrease in numerical rating scales, that would be 0 to 10. By 2013, the treatment advantage decreased. That is a p-value of 0.003. So 3 chances in 10,000 that this was not a real finding, with only an 8.9% decrease in pain over baseline. Now, contrary to what you might think, you would think a bigger study would get rid of some of the garbage and give you a more reliable result. But in fact, placebo responses increase with the sample size and the study duration. The worst differences by far were in this country. Now, as an example, in the early 2000s, you could still do a pain study for 5 weeks. There was an organization called Impact that recommended 12-week treatment. When that happened, results got worse. Again, this may be counterintuitive, but that is what has happened. Particularly with cannabis, being a drug that can produce intoxication based on THC concentration, it is always an issue as to whether patients feel it, and can tell the difference between what they're taking and the placebo that's supposed to be inactive. With Sativex, this is a liquid. The placebos that were used were a strong taste of peppermint, which was also in Sativex itself. So people can tell based on taste or the appearance of the material. In the studies, no statistical differences were observed in the incidence of euphoric mood among patients with prior experience of cannabis versus those who had never tried it. And this is very low to begin with, 3% in each. So the regulators liked this because it didn't indicate a high drug abuse liability. And it seemed there wasn't a big difference between placebo and the drug itself. There also were no differences in the two groups with respect to how well it worked in treating symptoms. So this would support the idea that studies were well blinded, that neither the examiner nor the patient knew what they were taking. Now, that's not the case for smoked cannabis. People most often can tell the difference because they're high when they smoke. Now, there was one study by Weir et al. Where they used very small amounts of herbal cannabis, 10% concentration, and they did single inhalations with a pipe. There, the blinding seemed pretty good. Um, and so it was enough medicine to show a pain response, but not necessarily to make people really high. Now, this was a problem in some of the Sativex studies. There was a situation where a prior study was statistically significant, except one patient who shouldn't have been in the trial because they were on a disallowed drug concomitantly. So this is a patient that was on placebo, and they had a marked decrease in their pain, spasticity associated with MS on placebo from beginning to end. Well, the explanation is they went into remission from their MS attack, and it had nothing to do with the drug. But that was enough to totally skew the data so that it was no longer statistically significant. In an effort to reduce this, the subsequent trial, the Novotny study that we see here, was done with what's called a randomized withdrawal design. Unbeknownst to the patients, everyone initially received Sativex. They were told at the beginning, you will get Sativex at some point. So after a few weeks, only the people who showed improvement in spasticity were then randomized. Half continued on the same number of sprays a day of Sativex. You'd see that in the blue or purple line. And half got the same number of placebo sprays a day. That's the orange or red line. And you'll see a marked divergence that led to statistically highly significant results. Why does it work? Well, might as well throw this in here, probably to be repeated in an MS module at a future date. Muscle tone is under tonic control of the endocannabinoid system. So CB1 agonists like THC reduce spasticity, while drugs like rimonabant that are inverse agonists that reduce endocannabinoid tone will exacerbate spasticity. The CB1 receptors are very densely packed in brain and basal ganglia areas, subserving motor control and corresponding areas in the cerebellum. Endocannabinoid functions are also very prominent in interneurons in the spinal cord that mediate muscle tone. As is seen here, cannabis-based medicines are clinically effective in spasticity and multiple sclerosis and possibly other conditions. What else can be done to reduce the placebo effect in randomized controlled trials using cannabis-based preparations? This is true in any trial. We need to limit patient expectations. You can't say, Mrs. Smith, we have this great new drug. I think you're going to find very helpful. That's going to skew the results because the expectations are lofty. Instead, one should say, this drug may or may not help you. Would like to find out. Patients need to be treated in a neutral fashion. Studies can go awry when there are too many ancillary benefits. Just to give an example of nice care, not good for a study, um, uh, there was a provider in Vancouver, Canada. When people came in for their herbal cannabis, they also had access to free massages, consultation with an herbalist. Wonderful place, but probably not a good milieu for a randomized controlled trial. It's very important to utilize a slower delivery technique. Again, inhalation is going to cause rapid peaks in the pharmacokinetics and rapid offset that's going to lead to risk of dependency. The preparation ideally should not be overtly psychoactive. So if it has THC, it's going to work a lot better with fewer problems if there's CBD, CBG, and a terpenoid profile that's going to buffer the THC effects. Now, we're going to see a long series of slides here related to the last federal study of the health effects of cannabis that was released in 2017. They do these about every 18 years, so it's going to be a while before we'd expect to see another one. This is from the National Academies of Sciences, Engineering, and Medicine. The data was cut off in 2016, just in time to miss a lot of important studies, particularly relating to cannabidiole's effect in epilepsy. So these are very conservative assessments by a panel of experts. To start out, substantial evidence that cannabis is an effective treatment for chronic pain in adults. This is based on evidence from all available studies, but insufficient evidence to support or refute the conclusion that cannabinoids are an effective treatment for cancers, including glioma. And we'll have more on that later. And that means primary treatment, something to treat the tumor itself. There's conclusive evidence that oral cannabinoids are effective antiemetics in treatment of chemotherapy-induced nausea and vomiting. So that goes clear back to nabilone and Marinol in 1985. There is limited evidence that cannabis and oral cannabinoids are effective in increasing appetite and decreasing weight loss associated with HIV/AIDS. So back in the 90s, this was a really important issue, but it was then that antiviral drugs came to the fore, and hopefully this is much less of an issue for people living with AIDS at this point. There's insufficient evidence to support or refute the conclusion that cannabinoids are an effective treatment for cancer-associated anorexia, cachexia syndrome, and anorexia nervosa. We'll deal with the latter later. The only available study was done 30 years ago with extremely poor methodology. There's insufficient evidence to support or refute the conclusion that dronabinol, synthetic THC, is an effective treatment for the symptoms of irritable bowel syndrome. There is insufficient evidence to support or refute the conclusion that cannabinoids are an effective treatment for epilepsy. That one's changed, and we'll address it subsequently. There is substantial evidence that oral cannabinoids are an effective treatment for improving patient-reported multiple sclerosis spasticity symptoms, but limited evidence for an effect on clinician-measured spasticity. What they mean here is measures like the Ashworth score have been difficult to show improvement, but it's not a effective drug if the patient doesn't feel that they're better. So which is more important in the end? There's insufficient evidence to support or refute the conclusion that cannabinoids are an effective treatment for spasticity in patients with paralysis due to spinal cord injury. There have been minimal studies. There is limited evidence that THC capsules are an effective treatment for improving symptoms of Tourette syndrome. However, Kirsten Mullerfall in Germany has done a great number of studies with other forms of cannabis showing a great deal of improvement without problems with neuropsychological testing. There is insufficient evidence that cannabinoids are an effective treatment for symptoms associated with amyotrophic lateral sclerosis. There's sure a lot of people with this disorder that are using it to advantage. And subsequently, as we will portray later, there now have been two phase 2 clinical studies, both with nabiximols, showing symptomatic improvement. There is insufficient evidence to support or refute the conclusion that oral cannabinoids are an effective treatment for chorea and certain neuropsychiatric symptoms associated with Huntington's disease. Not a big surprise. This is one of the most difficult conditions in medicine for any drug to produce improvement. There is insufficient evidence that cannabinoids are an effective treatment for the motor system symptoms associated with Parkinson's disease or levodopa-induced dyskinesia. Parkinson's disease is very difficult to treat as well. We will have a separate module related to this and current conclusions. There is insufficient evidence to support or refute the conclusion that nabilone and dronabinol are an effective treatment for dystonia. Again, good luck with any drug to treat these movement disorders. There is limited evidence that cannabinoids are ineffective treatments for improving symptoms associated with dementia. Again, there is a great deal of anecdotal evidence of benefit on agitation, sleep disorder, etc. And what we need is clinical trials there. There's limited evidence that cannabinoids are an ineffective treatment for improving intraocular pressure associated with glaucoma. Well, in fact, the first patients that got cannabis legally through the Compassionate Use Investigational New Drug Program, a couple had glaucoma and were able to show to the government's satisfaction that nothing else worked to reduce their eye pressure. There is limited evidence of a statistical association between cannabinoids and better outcomes, such as mortality and disability after traumatic brain injury or intracranial hemorrhage. Certainly, the basic science on this is very supportive of likelihood of improvement. There was a synthetic medicine called dexanabinol that looked promising in phase 2, failed in phase 3, but for whatever reason, the company that used this employed a single injection after the event and with no effort to treat on a continuing basis. There is no evidence to support or refute the conclusion that cannabinoids are an effective treatment for achieving abstinence in the use of addictive substances. Well, we're still trying. And again, the bulk of the data indicates that cannabis is an exit drug, is not a gateway drug, and can reduce the use of opioids for pain or addiction to other substances. There is limited evidence that cannabidiol is an effective treatment for the improvement of anxiety symptoms as assessed by a public speaking test in individuals with social anxiety disorders. This refers to a single study by Bergamaschi in Brazil, where 400 milligrams of pure cannabidiol did improve anxiety in this context. There is limited evidence that nabiximols, dronabinol, and nabilone are ineffective treatments for the reduction of depressive symptoms in individuals with chronic pain or multiple sclerosis. In the psychiatric module to come later, we'll address this in much more detail and show that there is evidence of benefit with the right preparations in the right context. There is moderate evidence that cannabinoids, primarily nabiximols, are an effective treatment to improve short-term sleep outcomes in individuals with sleep disturbances associated with obstructive sleep apnea, fibromyalgia, chronic pain, and multiple sclerosis. The fact is that along with pain, sleep disturbance is one of the most frequent reasons that people use cannabis medicinally. And a vast majority of studies where this has been assessed, patients show benefit. And again, this hasn't been formally studied with a standardized medicine aside from nabiximols, which has improved sleep in almost every study. There is limited evidence, one single small fear quality trial, that nabilone is effective for improving symptoms of post-traumatic stress disorder. This particular disorder has been extensively studied with NIDA cannabis, but none of the studies have read out positively, and I would attribute that not to poor study design, but poor material employed. Again, this is THC only material with no particular terpenoid content, so not an optimized agent. There is insufficient evidence to support or refute the conclusion that cannabidiol is an effective treatment for the mental health outcomes in individuals with schizophrenia or schizophreniform psychosis. Well, there have been 2 phase 2 clinical trials that would beg to differ. So after this came out, Caroline McCollum and I wrote our own version of cannabis administration and dosing and our assessment of where things stood. Um, these are different routes of administration that we'll hear more about later. The obvious: smoking, vaporization, oral, and other routes. I would just point out that with suppositories, the significant data to indicate that most cannabinoids are not well absorbed from the rectum. A synthetic THC hemisuccinate supposedly does better, but it really hasn't been developed. This is a comparison in how things work. Onset is quite different. Smoking or vaporization is listed as onset in 5 to 10 minutes, when in fact, with a high potency material, people will feel it instantly. It's clear that oral preparations might take 1 to 3 hours. For oral mucosal preparations, somewhere in the 15 to 45-minute range, so slower, faster than oral, but a lot slower than inhalation. Topical, another issue. Until recently, it's been extremely difficult to demonstrate significant serum levels after application of cannabinoids to the skin. They just don't get in well. Duration: smoking often lasts 2 to 4 hours, meaning that repetitive dosing is needed for treatment of a chronic condition. Oral's better, often in the 6 to 8 hour range, same for oral mucosal, and goodness knows with topical. So there are advantages and disadvantages. I do not recommend smoking for any indication. Vaporization is better, but again, for a chronic condition, most often oral or oral mucosal preparations are going to be the best approach. There could be a case for getting something in a hurry on treating an acute condition like cluster headache is one example where vaporization might be useful. This was our rendition of the level of evidence. It does adhere pretty much to what the institutes of medicine, et cetera, found, again, conclusive or substantial evidence for chronic pain treatment, multiple sclerosis spasticity symptoms, chemotherapy-induced nausea and vomiting. The big difference was treatment of intractable seizures in Dravet and Lennox-Gastaut syndromes. Subsequently, seizures associated with tuberous sclerosis is also an approved indication. Again, moderate evidence of efficacy in sleep disorders. Again, we think there's good evidence in treatment of glaucoma, increases in intraocular pressure. Um, Again, limited efficacy on a formal level for dementia, Parkinson's disease, et cetera, and on and on. Now let's talk about specific disorders and start with epilepsy. This has been controversial. THC certainly can cause seizures in rodents at extreme doses, but this has never been substantiated in humans. As a neurologist in my entire clinical career, I've never seen a situation where I thought a seizure was precipitated by exposure to THC or cannabis. There have been cases where somebody had what seemed to be a withdrawal seizure, but that's different. Cannabidiol is anticonvulsant, and it's also been shown for THCV, linalool, and likely THCA. Cannabidiol is approved as Epidiolex for severe epilepsy syndromes. The doses required are very high, typically 20 milligrams per kilogram per day or even higher, maxing out at 2,500 milligrams in adults, and you better believe that's expensive. Years ago, it was estimated that the cost to the patient who had to pay for it would be in the range of $32,000 a year for an adult. Similar or better responses were noted at a much lower dose, 8 milligrams per kilogram per day or less in full spectrum preparations. Dustin Sulek reported this, and there was a study by Pamplona that showed this clearly, although the patients getting CBD extracts were not in randomized controlled trials. Cancer. Now, there are two aspects here. One is treating symptoms associated with cancer. So clearly, cannabis preparations can be beneficial in nausea and vomiting from chemotherapy or radiation. There were two positive phase 2 randomized control trials of Sativex in opioid-resistant pain. The phase 3 trials did not read out. However, it did work in the American subspecies. It did not work well in Eastern European patients, particularly Romania, where the patients were much sicker. In those countries, it's very hard to get opioids for your cancer pain, and once someone had failed that, they usually were near death and nothing really helped. Again, insomnia associated with cancer. Now, the real holy grail is primary treatment of cancer. Cannabinoids, as we've heard, are cytotoxic for cancer cells, promoting apoptosis, programmed cell death that normally is turned off in cancers with immortalized cells. But the cannabinoids are protective for normal cells. Generally, large doses are required, somewhere between 500 to 1,000 milligrams a day of total cannabinoids, given long term. And it works better in association with conventional treatment. One truism is if someone responds and has reduced cancer or apparent elimination of cancer, that's got to be continued indefinitely. We have seen all too many cases of response followed by relapse when people quit the medicine. Now, the results have been better in treatment of glioblastoma multiforme, the most severe brain tumor with Sativex. And we see details on the study here. This actually was done some years ago and wasn't reported formally until 2021. Now, this was called a phase 1b randomized placebo-controlled trial. Um, it was with nabiximols or mucosal spray in conjunction with high-dose temozolomide, which is a standard chemotherapy agent. There were 21 patients who had recurrent GBM on the temozolomide plus Sativex or placebo. They capped this at 12 sprays a day, which would be 32.4 milligrams of THC and 30 milligrams of CBD. Many patients will tolerate that. It's a much, much lower cumulative dose than we've seen for most cancers. But gliomas seem to be very sensitive to cannabinoid treatment. So in the patients who got Sativex, 83% had a 1-year survival versus 53% in controls. So it was statistically significant. There was also a nice increase in survival time, 550 days compared to 369 in controls. And there wasn't a huge problem with adverse events, only 2 withdrawals in each group. Pain. About 70% of patients who use cannabis medically have some kind of pain condition. However, it's become abundantly clear that cannabis is poorly effective for acute pain, except perhaps in conjunction with opioids. So if you're having a molar extraction, you don't want to be high for it or after it. But in contrast, it's got a wide spectrum of activity in chronic pain, and that doesn't matter whether it's so-called somatic pain or neuropathic pain. By far, the best documentation is for type 2 preparations combining THC and CBD. Obvious example being where the most studies have been done with Sativex. And we'll have a lot more detail on that in another course. The elderly. In fact, Sativex was extensively utilized in elderly populations. I was the pharmacovigilance officer for GW Pharmaceuticals in the development program for many years, and we really saw no particular increased adverse events, side effects in older people as compared to younger people. It remains true with this or any other cannabis-based medicine that the titration, getting people onto the target dose should be very slow, with consideration of what other drugs they're on, and with good warnings about the possibility that too much THC could lead to sudden drops in blood pressure, orthostatic hypotension that will lead to falls and injury. Parkinson's disease. A wide variety of preparations have been tried with mixed records on treating the rigidity, tremor, and psychosis. The best study was actually a survey study from the Czech Republic, Venderov et al. And this was likely acid cannabinoids because it was oral use of raw cannabis after prolonged administration. So there's been no follow-up on this that I've been able to find. There have been various studies we'll go into in specific detail in a later course. As previously alluded, in schizophrenia, there've been 2 studies, but interestingly, a lot of psychotic patients use cannabis on their own. They also tend to be high volume users of other drugs of abuse, possibly attempting self-treatment. It is true that cannabis may precipitate earlier, more severe disease expression only in someone who is susceptible. No study has been successful in showing that cannabis creates schizophrenia where there wasn't the predilection beforehand. Again, cannabidiol at very high doses, 800 to 1,000 milligrams a day, show benefit on positive and negative symptoms in two phase 2 randomized control trials at doses of 800 to 1,000 milligrams a day. Pediatrics. It may surprise people to know that in the 19th century, when Indian hemp came to Europe and the United States in particular, cannabis was widely used in children and there were no particular problems. Children under 10 seem to be quite resistant to the psychoactive effects of cannabis. Um, Additionally, cannabidiol and tetrahydrocannabinolic acid are particularly useful. THC is proven to be quite calming in severe behavioral disorders associated with autism and other static encephalopathies. Paradoxical effect has been noted with low dose CBD in autism. Our friend Bonnie Goldstein recommends starting at 50 milligrams 3 times a day. And actually, this is a borrowed slide. This is a set of recommendations that she uses in treating children with autistic spectrum disorder. One of the issues is, can cannabis be combined with opioid treatment or in someone who seems like they're dependent? It is the case that cannabis can be used concomitantly with opioids, even in those who are addicted, but they should be closely followed. After introducing cannabis, if a patient is doing better for a time, then it's a good idea to try and reduce the dose of the opioids or taper them off of it. In practice, what we often hear from cannabis clinicians is the patients figure this out on their own. And if they're feeling logy or are constipated from the opioid, they'll begin to reduce it. It's not unusual for a patient on cannabis to come back in and tell the surprised doctor that they're off of their opioids entirely. Um, our colleague Dustin Sulak has a resensitization procedure. If a patient comes in on what he considers excessive doses of cannabis due to the development of tolerance, and they still need it to treat a medical condition, he will ask them to cut the stop for 2 days and then resume at half of the prior dose. Interestingly, pain control or other issues seem to be as well controlled on the lower dose. So, just to sum up again and repeat this, where are we now with cannabis efficacy? So we have solid clinical trial proof for cannabinoid treatment for either cannabis-based medicines, THC or CB1 agonists for nausea and vomiting, anorexia associated with chemotherapy, HIV/AIDS, spasticity and multiple sclerosis, and likely other neurological conditions, neuropathic pain, whether peripheral or central, cancer pain, lower urinary tract symptoms, and glaucoma, although there have been no recent studies. For cannabidiol, intractable epilepsy with these 3 seizure syndromes, for which there are multiple phase 3 randomized controlled trials. And then again, schizophrenia, positive and negative symptoms, and 2 phase 2 randomized controlled trials. Another tangent, problems in cannabis laboratory analysis. In fact, most labs doing this work don't have Schedule 1 permits, and technically what they're doing is illegal. There's no rule book that everybody has to follow, so we have lack of uniformity in the methodology. Labs sometimes cheat. There may not be good due diligence, and they may engage in what's called dry lab results, where somebody would look at the cannabis flower bud and say, well, it's got a lot of trichomes, so it should be 25% THC, obviously made up. There sometimes is not a good availability of cannabinoid standards for all the different cannabinoids. It's true that cannabinoids are tough to assay properly, and the terpenoids in lower concentrations are even tougher. And there are only certain specialty labs that can assay pesticides properly. Looking at needs for the future in basic science research, we need to do better breeding, understand the genetics and genomics of cannabis better, um, ascertain what the metabolic and pathways are for cannabinoid and terpenoid production, figure out best component ratios, and have a situation where seed commerce is allowed. Other issues, organic culture versus genetic modification, pesticides, heavy metals, uh, microbial contaminants. We also have a severe knowledge deficit in medical school where not only is cannabis not taught, the endocannabinoid system, despite its prevalence and major effects on physiological processes, isn't taught either for the most part. And continued opposition, whether it be insurers, medical societies, or politicians. What do we need in terms of prospective cannabis research? Uh, we need access to standardized, good manufacturing practice cannabis with appropriate cannabinoid terpenoid profiles, genuine phase 2 and phase 3 clinical trials that satisfy the FDA for pharmaceutical development. And same kinds of standards would apply to over-the-counter products, whether they be topicals, skincare, cosmeceuticals, etc. What we really don't need are a lot more case studies or generalized surveys. Yes, we've done them. We've done them sometimes because that's all that was allowed. We don't need more NIDA-supplied studies that can't be reproduced or advance the situation of research knowledge. And we don't need to be wasting tax money from the public. These are some suggestions on clinical research priorities. Obviously, chronic pain is extremely prevalent throughout the world, and particularly for unstudied conditions. This is a very promising area. Arthritis, both rheumatoid and osteoarthritis, the inflammatory bowel diseases, and then, on the other side of the coin, using THCV and CBD to treat the metabolic syndrome and insulin resistance. Dermatology is readily accessible with little to no risk of side effects to deal with acne, psoriasis, and contact dermatitis. Certainly, we're very interested in neuroprotection, whether it be due to dementia, traumatic brain injury, stroke, or the like. We should also be thinking non-drug approaches. What can we do to optimize endocannabinoid system health? This would include lifestyle and nutritional research. These would reduce cannabis need. And then we've got the forbidden territories, particularly psychiatry and OBGYN, where there's been so little work done. Conclusions. Cannabis has proven medical potential and has led to the discovery of the endocannabinoid system, a major physiological homeostatic regulatory system. Cannabis in the proper formulation can become an approved pharmaceutical in the United States and elsewhere, fulfilling necessary criteria of safety, efficacy, and consistency. Well, I don't have a crystal ball. A lot of what happens from here depends on political events, whether additional research is possible, whether cannabis is legalized in any form. But there's great potential for advances in cannabis therapeutics, but also fiber production, hemp seed nutrition, and production of cosmetics. And with that, we'll close. I will stop sharing.
Thank you so much. So since Cannabis and the endocannabinoid system isn't taught in medical schools. We have a lot of people who are trying to treat themselves with cannabis, and many of whom who go on to PubMed and like to look at scientific articles. So, what advice do you have for the non-medical person who is trying to learn from the existing research in terms of what should they be looking for practical application, and what cautions do you have for them?
Sure. Well, the ideal situation is to have the advice of a clinician that's well-versed in cannabis-based medicine. They could find wherever they live, they could plug their zip code or location into a search engine at the Society of Cannabis Clinicians to find a practitioner that has passed a test to show competency in these issues, and we'll supply that URL to people. Beyond that, self-education in this area is very complex. Certainly, what we would offer is that any of my articles, including all the material in this lecture, would be available free uh, with access at ethanrusso.org at the library tab. So I certainly encourage people to go there and find out what is of interest.
Will you explain the importance of the peer review system for scientific publications?
Sure. So when someone has an article about cannabis or anything else that they want to publish in a scientific journal, it is submitted to the journal and it's supposed to be reviewed by two or more experts that will critique and suggest corrections, or they may reject it out of hand. The idea behind this is to try to ensure that the study was done properly and that the results are not misrepresented. We can't always know if someone's cheated or fudged data, but it is an effort to make sure that the data is accurate and worthy of publication. Now, I will say that it's a very tough system. We see a lot of garbage that's published that one questions whether it went through any kind of peer review. On the other hand, some great material doesn't get published or gets published only after it's gone to 4 or 5 journals. Um, some journals have specific criteria of what we're interested in this month, and if it's not, it could be Nobel Prize worthy research, and it may not make the cut because of other factors. So it can work both ways. An additional factor is nobody wants to review these studies anymore. I function as an associate editor for a cannabis journal, and it takes months to get the requisite number of people who agree to review the material. So it's very difficult. Again, things can go smoothly or they can go very badly in this regard. But the intent is an honest one to try to ensure that useful scientific advancements get published and things that are questionable do not.
What are epidemiological studies?
Epidemiological studies look at a population and what the incidence or prevalence is of a disorder. With respect to cannabis, commonly these studies have to do with harms. So can you show that people who smoke cannabis are at greater risk to have heart attacks, that kind of thing. But similarly, there are surprises. People intuitively think cannabis produces the munchies and must make people fat. But, when this was actually looked at, chronic use of cannabis have a lower lean body mass. So that really made people think. There are reasons for that, and we'll sure talk about that subsequently.
In the context of clinical trials, we often hear phase 1, 2, 3 studies. You mentioned phase 1B study. What's the B indicating?
Well, the B could be different things. It means there's been some preliminary work. They might have thrown in a few patients that had the disorder in question, so it wasn't just normals who got it, that kind of thing. Yeah, so it would usually indicate that it was a little more focused on a specific disorder rather than just seeing what happened with folks with no particular issue medically.
You've probably been involved in more clinical trials with botanical cannabis drugs than maybe anyone. What drug abuse liability issues have you seen in those trials?
Well, again, we've seen that there's been a gradual realization that there's less of an issue than people feared. We talked about how synthetic THC went from being a forbidden drug to Schedule 2 and then Schedule 3. Um, Sativex isn't approved in the US. I'm not sure where it would have ended up. Um, a really good example is cannabidiol. Although it's not a controlled drug in most of the world, in the US, it is considered a Schedule 1 forbidden drug, except in the context of Epidiolex. So Epidiolex was initially put in Schedule 3, meaning that that's supposed to be drugs with some potential for addiction. It had to be closely monitored. Subsequently, with study Babylonis et al. Showing no drug abuse liability to speak of, with cannabidiol, it was descheduled. And there now are discussions underway in the US about downscheduling cannabis from Schedule 1, forbidden, to something else, or even descheduling it. But we're waiting and waiting and waiting.
So you and I are involved in a variety of botanical cannabis drug development projects. Will you talk a little bit about why so many of the drug development deals that start off wanting to make something that is a botanical drug transition to manufacturing a synthetic cannabinoid or synthetic cannabis-based drug?
Yeah. Well, the standard paradigm that drug companies follow is they want to have full intellectual property rights to things, and that means it's their own creation. So typically, they will design a drug um, that lodges on a specific receptor with the highest potency and affinity and go from there. Unfortunately, it's sort of backwards because um, it might lodge on the receptor, but they don't pay enough attention upfront to the possible toxicity. The result is a lot of drugs are initially approved and then come off the market when they're given to bigger groups of people and we discover that they're bad for the liver or they poison the bone marrow or something else. With cannabis, we've got a very different situation. We know that this substance has been used by humans for thousands of years, and in general, nobody dies from it. Um, it is a situation where there's been a lot of hysteria attached to cannabis. Um, uh, on the botanical side, as I pointed out, getting an extract of a botanical and having it be the same from soup to nuts from the start of the program through approval by the FDA, it's a daunting task. However, it's been done and it can be done. Um, Again, some of us have a bias that cannabis as an extract is going to be more effective and safer than any semi-synthetic or synthetic drug that may have just a very narrow um, activity against the disease, but not treat ancillary symptoms. Um, in a manner of speaking, cannabis is like polypharmacy in one preparation because different components can contribute to symptom reduction, as well as try to treat the pathological process involved in whatever the disorder might be. So there's great potential there. Instead of grandmother taking 15 drugs, it may be that a properly constituted cannabis-based drug can provide everything that she might need,
Thank you.
ideally.
Right. Well, let's talk a moment about the National Academy of Sciences report. So it seems that um, you think that they may have missed the mark a little bit in a couple of areas. And hopefully the next report will be better than the last one. But I'm curious, is you mentioned this is the last report published by the government on this topic. So are the people who are involved in putting this report together government employees? Are they from medicine? How did this report get put together and who is involved?
Yeah, it's a combination of clinicians and basic science scientists, from American universities for the most part. The people were well qualified. It really didn't include, let's say, doctors in the trenches treating people with cannabis-based medicines. So, it had to be based on literature reviews, and obviously, because of the hierarchy of proof, there was a strong reliance on randomized controlled trials where they existed. They had to assess the levels of proof as we discussed initially. So I'm not criticizing them, but in a rapidly advancing field, having this kind of assessment every 18 years is clearly inadequate. So things have changed so much just in the last 7 years. Well, eight really, because they cut off data in 2016. They've got to reassess. The problem is the decisions as to who gets it and how still often rely on politicians who are really in a poor position to be the arbiters of what's good science and what the changes in policy should be.
Thank you. My final question is about insurance. Do you think there's any point in our future when we'll see insurance companies covering the cost of cannabis and cannabis preparations?
They will if they're FDA approved. Beyond that, I think that there's been reimbursement for medical use of cannabis in Germany it was at least a consideration in Spain, but for the most part, that hasn't happened. Um, Often to do that, the governing body will have to show that there's a cost savings. For example, with Sativex, there was a study showing decreases in cost in physical therapy treatments by virtue of treating spasticity in MS. Um, so again, uh, take into account our cost benefit analyses. I will be cynical but accurate to say that American insurance companies are going to do their best not to pay for anything. So they're very good at finding excuses not to reimburse for a given condition or a given medicine.
Well, if I remember correctly, it was a father-daughter research team, maybe Bradford et al. Is that the
Yeah,
correct name? That took a look at how much less people were spending on pain medication on Medicare Part D. Do you want to talk a little bit about that before we conclude?
sure. So they looked at in separate studies Medicare costs and use rates for opioids in states that uh, became where cannabis became available medically. And the results were very compelling. There have been a series of studies showing similar findings, and one that purported to show that things were worse, but I none of my friends or colleagues believe it. No, I mean, those involved in the field know very well that cannabis-based treatment will lead to decreases or even discontinuation of opioid treatment. Now that it's been well established that opioids really aren't effective in non-cancer chronic pain, we need to be using more cannabinoids that do work and aren't going to kill people due to overdose. Things can be better.
Here, here. Well, thank you so much for an excellent presentation.
Thank you.
We'll see you guys again soon.